The same religion that's capable of hideous acts of destruction can also be capable of moments of healing, of restoration, and of hope.
But educate a girl, and you educate her entire family.
There is a sun within every person.
When that anger sets in, write it. Write the letters, but don't send them. You never want to leave concrete proof of insanity.
Sometimes you can't quite say enough about a person. You can't, no matter what you do, you cannot introduce them properly. Troy Duster is someone for whom no introduction really can do justice. Perhaps the leading sociologist of race in the United States. He's been on the cutting edge of critical race studies and justice studies as well for so long and so effectively that he has remade these fields of study in many, many ways. And he's deeply influenced activism, activist social practice as well. And as someone whom I've known for quite a few years, I can testify to his influence. Although I was never formally his student, I've been his student in a deeper way since I think about 1970 or so. So we go back. Today, as we encounter sometimes confusing trends in the development or the resurgence of research claiming to be doing scientific work on race, scientific studies, it's good to have Troy Duster's insights on hand, because Professor Duster is unquestionably the world's leading authority on race and science and on scientific racism. I'll just say a quick thing about Troy Duster's qualifications and let people come to take seats for a moment here. Come on in, please sit down. Okay, thank you. Troy Duster is Professor of Sociology, Director of the Institute for the History of the Production of Knowledge at New York University. He is also Chancellor's Professor of Sociology at UC Berkeley, where he served with distinction and chaired the Department of Sociology over many years. He served as president of the American Sociological Association in 2005 and received one of the highest awards that the sociology profession has to bestow, the Du Bois Johnson Fraser Award in 2001. He's the recipient of numerous other awards and honorary degrees. He's the author of Backdoor to Eugenics, whose second edition, very, very relevant to the discussions we'll have today, came out in 2003. And he's also the co-author of Whitewashing Race, The Myth of a Colorblind Society, among numerous other publications. It is a great pleasure and honor for me to present Troy Duster today.
Thank you, Howard, for a generous introduction and for the invitation to speak. Actually, Howard Winnott is one of the leading figures in the study of race around the world. This is a reflective response. Let me try to put into context my title, which is a bit counterintuitive. Why? Well, what can the DNA tell us about race? Up until 2000, the answer was nothing. The mantra was, at the DNA level, we are all the same. the first decade of the Human Genome Project was characterized by a particular formulation. It went something like this. Doesn't matter whose genome you sequence. It can be Aboriginal, it can be a Laplander, an Asian, a white person from Europe. Anybody's genome will do for the map. Why? Because our DNA is so universally homogeneous, it just wouldn't matter. So what happened in 2000? Well, there was a press conference at the White House, and Bill Clinton and Tony Blair, then and still Prime Minister of Great Britain, got together with Craig Venter and Francis Collins. At that press conference, they made a statement. It went something like this. We have our disagreements, they said. Venter, after all, was the head of the private sector of sequencing of the genome, and Collins was head of the NIH DOE public sector. But one thing we agree upon, they said, at the DNA level, there's no such thing as race. Okay, so let me step back and provide some historical, social context for my remarks. If we go back to the middle of the 19th century, and for about 100 or 110 years, we can locate around the Western world what Lakatos would call research programs designed to show that racial taxonomies mapped onto human biological processes or human characteristics. As early as about 1855, there were people in medical fields publishing articles about how the lung capacity of blacks and whites was systematically different. Later part of the century, Lindy Braun's work and also that of Evelyn Hammonds at Harvard, they're both publishing in the area, and they show how this framing, which comes out of course slavery, became part of the science establishment. And for a full half century, work went on, heavily funded, to show that blacks and whites had very strong differences with respect to lung capacity. Early part of the 20th century, the ABO blood system was used by the French and the Germans, and there were research programs trying to show that you could map onto the ABO blood system racial taxonomies. 1930s, the theory of fast twitch muscle emerges. That is, that somehow racial groups have different muscle density, and that you explain why Jesse Owens won the Olympics because of blacks' thicker density of fast twitch muscle. In some ways, the Tuskegee experiment, placed in this context, makes more sense. Because coming out of the 19th century, it was common sense in scientific work that blacks and whites were fundamentally different in terms of certain human physiological or biological processes. So when Tuskegee comes along, it's not merely to be dismissed as a kind of deeply racist version of the body. Some of the more progressive parts of the public health service were saying, well, if we let this experiment go on, we will show our colleagues that syphilis comes through the black body in the same way as the white body. Because before syphilis, there was a debate within science that syphilis would be different in whites than blacks. So we moved on into the 50s and the 60s, and something happened. With all the programs I just described, each one of them failed. Not a single one was able to show any pattern difference between taxonomies of race and human biological, physiological processes. Not the blood system, not the capacity of the brain to absorb or respond to certain kinds of neurotransmission patterns. There was nothing that mapped along race. By 1970, you could not find a single research program funded by the federal government and almost none by the private sector. The Pioneer Fund was one of the exceptions here, which kept this line of research going. And by the 1980s, at the dawn of the Human Genome Project, the mantra was, maybe we should just get rid of the concept of race. And so my first slide is a kind of ratification of this. Here's a consortium of scientists publishing the second UNESCO statement on race. And notice the high drama, the unequivocal statement in 1995. It says, this concept of race has no place in science. That's a consensus statement. This is before the Human Genome Project. So there was this internal coherence, consistency about the biology of race. And you can see the title of the article is Race a Dilemma Concept for Science? And the answer at this point was, of course, no. The same year that Blair and Clinton are saying it's all over, there actually are plenty of seats here, so rather than standing in the back, why don't you take one or two? At the same time that Blair and Clinton are saying it's all over and the scientists are agreeing, at least on the public stage, something is going to happen that's going to help shift the nature of this problem. The Congress passed the Minority Health Act, and best of intentions, the idea was to chronicle, to detail, to give accounts of health disparities in the hope that we could focus upon how to eliminate them. So the Congress tells the NIH to support research on disparities between racial ethnic groups. And here's what began to happen. Once scientists in these fields began to report out data showing that there were disparities between different groups, they didn't have the capacity to do other than interpret analytically that the differences that they were reporting must be accountable in terms of the racial taxonomy. So here's an example. Black asthmatics displayed diminished glucocorticoid responsiveness compared to whites, and the differences were significant. Conclusion? Well, concentrations of glucocorticoid in vitro suppression suggested blacks have a predisposition to diminish responsiveness. Now that's just racial predisposition. That could be neutral. But notice now what the conclusion said. Suggested blacks have an inherent pro-inflammatory cytokine. Whoa. This is the synthesis journal of the chest physician. This is not 1880, Lundy Braun's work. This is 2005. An inherent difference? This is not contingent. This is not a complex statement. It's saying it's inherent. I spent several years on the National Advisory Council of the Human Genome Project. And in that capacity, I met people at the Food and Drug Administration and the NIH and the National Science Foundation, and developed friendships, and some of them became deep throat. That is, they will send me letters. They'll say, Mr. Duster, you'd be surprised at how things have developed here. And here's a letter from one of my deep throats. This is an approval letter to Merck for the new drug, Cozar. So you can read it. Reducing the risk of stroke, left ventricular atrophy. And then it says, indications note that we noted. No benefit to blacks. Again, notice the lack of contextual analysis, the contingent, the notion of the possible nuances. No, black patients just don't benefit. Now here's an annual review, post-2000, post-genome, pharmacology and toxicology. Annual reviews, as many of you know, are not simply articles. They are distilled versions of a whole field of a whole year. So this is not cherry picking, this is what they concluded. Understanding the molecular basis underlying ethnic differences in drug metabolism, we need that to get to individualized drug therapy. Fair enough, but what do they mean? Well, what happened to individuals? In this account, it's ethnicity. And now white patients require higher doses of warfarin than do Asians. And even more specifically, Chinese require 50% lower average maintenance than whites. Now, ethnic is in the title, but in no way that I understand anthropology or sociology or any other discipline is white and ethnic. That's a racial category. And that you see throughout the fields of inquiry. And now I'm going to shift from it and then come back to what's behind this. This article appeared, again, post-genomic, inferring ethnic origin by means of a short tandem repeat profile in the DNA. Forensic science, and we're going to report the use of DNA profiles. And now here's the actual language. You can't read it. This is the actual language. The method is the estimation of 5 British populations. Notice again the title, ethnic origin. But Caucasian is hardly ethnic. That's a racial category. Afro-Caribbean, Indian subcontinent, these are the taxonomic systems in play. And they're going to now infer ethnic origin looking at the DNA. What's the purpose? To report ethnic origin using these sampling procedures. To get the probable ethnicity of an unknown offender. To assist in targeting for investigations, setting priorities for mass screens. So we're now going to use the DNA to limit or increase our capacity as dragnet specialists. We report the DNA profiles of these 6 repeats in the DNA to infer ethnic origin of a crime stain. Okay, now wrap your mind around that phrase. The ethnic origin of a crime stain. And discuss how this inference can be used to reduce the number of people to be interviewed and to resolve a case. Now, at that point, they only had a very small database, and they say as the loci are analyzed, we get more and more of them, a more powerful tool for inferring ethnicity. But read race. Now things get interesting. A couple of years later, this case appeared. Serial rapist, murderer. Went on for about 2 or 3 years, couldn't find him. An eyewitness had thought that was a white male. And so they had done a dragnet in that area for over 1,000 white males. But they had not succeeded. And then DNA print genomics, you'll hear more about that in a moment, came forward and said, you know, why don't you give us some of the data? Give us a tissue sample from the crime scene, and we'll try to infer ethnic origin of a crime stain. Prudhomme said, you're looking for a needle in a haystack, but it's the wrong haystack. Give us the tissue sample, we'll do the analysis, and we predict 85% African ancestry, the rest Native American. Bingo! Voila! Turns out they struck pay dirt. That's the person whose DNA ultimately matched, convicted, and sent to prison. Now, on the DNA Print Genomics website, if you go up there, they'll tell you about this case in rich detail, and then they'll say that police departments around the country should come to them for assistance. That they'll be able to help police departments using tissue samples left at crime scenes to reduce the number of people to be reviewed in the dragnet, and the issue is going to be race. How do they do this? Well, this is privately held software. So the answer is, we don't know specifically width and depth of sampling, but here's what we know from their own analysis, that they're sampling very broadly in these 4 or 5 zones. They're getting a few, let's give them credit. Let's say they're sampling as many as several thousand people, and Native Americans, different zones. And then they're saying, if you can find sets of markers that seem to be more distinctive in groups A than group B and C, then that'll be your, quote, sample size for that whole population. Now, I won't get technical here. At a certain point, I'm going to turn to what I really think of this technology. But before I do that, let me show you where this is headed. If you go up to their website, Ancestry by DNA, which is another version of print genomics, they'll tell you if you send in a sample of your saliva on one of these computer chips, they'll send you back in a few weeks percent ancestry. What percent are you that's white, black, Asian, Native American? Sound phantasmagoric? Not really. So some students at Penn decided to do this in a classroom, and the New York Times made it a front page story. I want to call your attention to the headline. DNA tells the students they aren't who they thought. Then the results come back, and students are, quote, are surprised. Professor Shriver, who is the key architect of this technology, DNA print genomics, the same company that did the work I described to you earlier in the successful forensic case in catching the murderer, the rapist. So what are they doing? Well, it starts off, they're interested in genetic mixing populations. It's about ethnic and racial backgrounds in drug trials. That was the purpose of the original work. However, researchers are also concerned about ancestry. So, Natasha Best gets her results back. Thought of herself as being half black, half white, but the test proved her to be 58% European, 42% African. What I've called in other contexts a recurring seduction to a false precision. But nonetheless, comes to believe that this is actually her ancestry. She's shocked. She thought she was half and half. Well, it's entertainment until it gets to money. Now, the black Seminoles have been around for a while, and it's not a big issue until there's a settlement. And there was a big settlement a few years ago. And the question, of course, then, well, who's entitled? Who's a Seminole? Well, 2000, the Seminole Nation of Oklahoma decides to make the constitutional amendment, 1/8 Seminole blood. What's that? And how do you know you're 1/8 Seminole blood? Well, usually that means grandparents, a particular connection to the past. And it's a little bit slippery. Should the Seminoles use DNA testing? And what would it mean to use DNA testing? Gets even more antsy. Last December, the Hard Rock Cafe, the whole chain, was purchased by the Seminole Nation for $1 billion. Now things get serious. Because now the question is, what are you going to use to adjudicate who you are? Is it going to be some records about grandma and grandpa? Is it going to be a test of your blood or a test of the DNA? This gets to be serious. And you may recall about February 1st and 8th of 2006, Skip Gates gets into the act with 8 famous African Americans: Whoopi Goldberg, Oprah Winfrey, Quincy Jones, there's Skip, Sarah Lawrence Lightfoot, T.D. Jakes, all very successful black Americans. Well, what's the issue? Black Americans, many of them, have simply taken the view, well, that what's past is past. But many black Americans have a certain kind of a quest, a thirst, to go back to find out, quote, who they are. That's because Middle Passage is unique in the history of African Americans. During the slave trade, all you got was a number of people on the boat that didn't give their part of Africa they were from. It just said a certain number. It gave it sometimes a gender or sex, but it didn't say anything about location. So the first 3 parts of this 4-part series by Skip Gates is relatively good in that it goes back through genealogical research to find out whose parents and grandparents and so forth were of all of these famous people. However, it gets to the middle passage, and there's a big black hole. What happened? And then Skip then turns to DNA print genomics. Mark Shriver in that group, Rick Kettles, and what does he ask? Can we use the DNA to go back into Africa and find out where we're from? And in a somber interview with each of the members, he then tells the people, and Oprah is told that she's not Zulu. She's been saying for the last 5 years, she knows she's Zulu, she looks Zulu, she goes to Zulu Nation, they welcome her, they kiss her, she knows she's Zulu. And then the DNA says no, and she's crestfallen. Sarah Lawrence Lightfoot, my colleague at Harvard, has been thinking all these years she's part Native American, and she and several others are told not a single African or Asian, pardon me, Native American bit of your background shows up in these tests. Now, what's interesting for me as a social analyst of science is the authority, the way in which each of these persons accepts the news as definitive. In the same way that the Seminole Nation could, if they wanted to, take the DNA and treat it as definitive, I'm now going to show you what I think some of the problems are. Ancestry by DNA and Ancestry.com are making some money here, $140 million annual subscriptions, so this is big money. If you saw the New York Times front page story about 10 days ago, there was a case of an immigrant from Africa who was trying to get his children into the country. And you have to prove that they're your children. And he went down the road of DNA testing. And it's a miserable story because it turns out that his wife had died, so she wasn't around. And then they did the DNA test on his children in Africa. Turns out that 3 of the 4 of them were not really his kids. You know the Maori stuff, well, who's your daddy? Well, you have these paternity suits, and it actually turned out, it's a terrible story, that this person's wife apparently had not been faithful based upon these DNA tests. So now here we have the social reality of having brought these kids up, adjudicated by the DNA. The DNA tells the government and not his kids. They were his kids, that he raised them, but they weren't really his kids. So they can't get into the country. See, this gets serious under certain conditions. Now, also in this frame, think of the following. We had the report in the newspapers about 3 months ago. Someone had done some studies of the English and the Irish, their DNA, and they found that they were remarkably similar. And as you know, there's been trouble in that part of the world. What might have been the purpose? Well, to adjudicate the social reality with the DNA? I don't think so. 10 years ago, there was a study about Arabs and Jews done by some Israeli molecular biologists. They were contrasting the Arabs and the Jews in their DNA with that of people from Wales. And they found that the DNA markers were much more close between Arabs and Jews than either to the Welsh. At the time, they might have thought that we're going to lay down their arms and Arafat would hug Sharon. It didn't happen. So the DNA adjudicates under certain conditions, but it is monumentally naive to believe that the DNA is going to have this effect. So, this is the famous case of the drug for black people, Bidil. Illnesses that we thought were actually dissimilar may have gene pathways. And so what we thought were differences between blacks and whites, diabetes, cancer, stroke, we thought maybe they were racism. Now it's nitric oxide deficiency. So there's consequence to this formulation. This is a famous paper, which I called to the attention of all my colleagues in anthropology and sociology who are asserting that race is a social construct. So here comes the genetic version of the counter. Gene clusters of the macrosatellite markers produced 4 major clusters, which showed a near perfect correspondence with self-reported race ethnicity. Astonishing. Indeed, when Stanford reported it out, they said, this is such a profound development that it challenges the notion that race is merely a social construct. Science. It has gene implications because they found the patterns in the microsatellite markers. Okay. Race is often said to be not a scientific construct, it's socially constructed. That's what we keep hearing in our courses in anthropology and sociology. And I think there's a misinterpretation of what this means. People say things, well, if it's socially constructed, therefore what? It's not real? Well, money is not a phenomenon of nature. You know, the Japanese have their yens, we have our dollars. You know, once upon a time there were marks and francs, now they're euros. In the last 10 years, you go from francs and, you know, pesos to euros. Obviously, socially constructed. Does that mean it's not real? Well, if you believe that, please pass all your $20 this way. In other words, the construction of the phenomena is real in its consequences. You get food and water and shelter with your euros or your dollars or your yens. In Argentina, 8 years ago, that went zoom down. Obviously, it's a social construction, but dramatically real. Now, the same is true for race. Constructed socially, but in Philadelphia, housing loan study, you actually can study this scientifically. Systematic empirical investigation of the consequences, and you find that people of a particular racial category have greater access to bank loans than others. So, money is real in its consequences, and you can study it scientifically. No one says, well, money is socially constructed, therefore you can't have the science called economics. They get a Nobel Prize for studying the consequences of constructed reality. Race. Now to the point of what I think about these technologies. This is a slide from Steve Olson. Steve Olson is has written a very good book on human genetic variation, and he uses this slide to show he can go back 10 generations, his own personal life. Why? Because in that part of the world, in Scandinavia, they keep great books, and they keep great books based upon patrilineal descent. Now you notice on the left side of the graphic, it only goes back 4 generations. They don't keep good records on mother's mother's mother. So why the claim he goes back 10 generations? That's because of something called social, political, anthropological issues, namely kinship structure and lineal descent. You keep records this way, and you can say this was actually your great-great-great-great-great-grandfather. Now, what's the importance of this for DNA? Well, we can now go on to the Y chromosome and determine whether or not Harry Olson is actually Steve Olson's grandfather, or whether indeed Frank is Steve Olson's father. The Y chromosome analysis is what permits us to know definitively who's your daddy. Now, you go back these 4 or 5 generations, and this is how they got Thomas Jefferson and Sally Hemings. Y chromosome on the Hemings side turned out to have the similar structural pattern of the Jefferson family. Now, for 150 years, scholars were saying, Tommy wouldn't do that. It's just not possible. Along comes the Y chromosome, DNA analysis, and all of a sudden the historians who had been saying it couldn't happen, they recanted faster than Galileo. They said, well, the DNA says so. Tommy must have done it. Our former president said, no, I didn't. The blue dress shows up, and suddenly we have a hearing, and we have him on tape. So the DNA starts to get pretty definitive. Let's move over to the other side. Turns out that the DNA, well, on the mother's side, it's called mitochondrial DNA. You can trace mother's mother, all the way back with this kind of analysis. You give me your DNA, and I can tell you whether or not your grandmother is actually your grandmother. Mary Claire King, research on the disappeared in Argentina. 20,000 people disappeared during the Dirty War. They had children. Many of them were 2, 3, 5 years old. The Argentinian military and their friends often adopted those kids and took them away. Years later, when the military fell, Mary Claire King goes down to Argentina and does mitochondrial analysis on the DNA of the grandmothers and the granddaughters, makes the link, and says, these are really your children. So here's the lesson. We can, with DNA analysis, do two things which are definitive. We can find out, on the mitochondrial side, on the female side, as far back as we have the data. So if we go back into the graves, we can find out all these people. Mothers. 10 generations if we could find the DNA. We could find the same thing on the father's side. Here's the trick. Each one of these people, indeed each one of these people, is equally related to Steve Olson as each one of these. In terms of the DNA, in terms of the actual DNA contribution, these are all equal contributors. Genetically, it makes no sense to say that this is your lineage, because this is also your lineage. You go back 3 generations, you're up to what? 1, 2, 3, 8, go back 4, up to, so you can count. 16, 32, 64, 128, 256, 512, and so on. By the time you get back here, you've got over 2,000 or 3,000 people who are all your equal contributors to your DNA. So why do you call this person your lineage? Why? Anthropologically, sociologically, politically, it makes sense. Genetically, it's nonsense. And yet we say we have the DNA lineage. Now we come back to the Seminoles and Oprah. Should the Seminoles use this test? Well, they're skating on very thin ice if they do this. Now at stake are millions of dollars. And I take a test, and I say, okay, I can go back and find that this person actually was on the Y chromosome, Seminole. And so far I make a claim. What you miss is everybody in between. The DNA cannot determine anyone else in your ancestry besides the 2 extremes on the Y and the mitochondrial. So if you're a Seminole, you'd be very careful about this series of tests. You'd make a huge mistake. Oprah, Zulu. What does it mean to say that someone is not Zulu? What does it mean to say someone is not Zulu? It means you'd have to have some understanding of all of this. Do we have the records on Oprah that go back like this? Categorically no. Those tests cannot be retrieved. And yet people nod their heads in assent. Okay. This famous graphic is the double helix, a series of nucleotides, just 4 of them repeated over and over again. You've seen this at least 1,000 times in your college career. What's different about this graphic is that it's cut open. It's cut open at the bottom. We could begin to do this about the year 2000. The same time we're saying it's all over, that race is all over, here's what happened. If you go through the genome of any two people in this room, 1,300 points, there'd be exact same overlap. These things, C, G, T, C, and so on, just like that. It's exactly the same. And then at about 1,000 points, there'd be a single nucleotide, these are nucleotides, difference. Those are called single nucleotide polymorphisms. For the next 1,000 points, exact overlap, any two people in the room or in the world. Go on through the next 1,000, 2,000, 10,000, 3 billion base pairs, same story. Any two people, the overlap would be overwhelming. 1,300 points approximately, and then a single point of difference. Now, here's an exact replication from Francis Collins' lab. It's courtesy of someone with these three nucleotide differences. On this chromosome at this point, pick out anybody in the room, put yours up there and match it, and you'd have the exact same overlap, except for these 3 points. So why is that significant? Well, maybe one of those 3 points is in what's called a coding region, and it would be, quote, a misspelling. It would tell the blood, let's say, to not clot when it meets the open air, in which case that particular polymorphism shift would mean that you would have hemophilia. So a single difference can make a huge difference in your life. But those differences were not coded racially. They're coded for something else. In the year 2000, despite this overwhelming similarity of 3 billion base pairs, let me pause here to make the point again. Any two people in the world, complete overlap at all these points in the nucleotides. But that means that any two people in the world have 3 million points of difference in their SNP patterns. They're called SNP profiles. Now the plot suddenly thickens, because in 2000, supercomputer emerges to solve the human genome's mysteries. And right at that point, IBM could tell you that they could calculate on the DNA 7 trillion per second calculations. Now what was only theoretical in 1998 becomes the source of a new field of inquiry called pharmacogenomics. And look at the first statement that was made in this field. The first statement made in science was that all of these SNP patterns differed in frequency among ethnic and racial groups. How did they know that? How could they know that? But the claim was made that this diversity meant that race must be considered in studies aimed at genotypes and phenotypes, and the purpose was to get different drug toxicity risk. That was the purpose. So a field is born. A few years later, this is the most famous paper reinscribing at the molecular level in scientific thought race. This is the paper that everyone refers to. Gene structure of human populations. Self-report is a good ancestry proxy. And here's what they said in the paper. They never mentioned race. The Rosenberg paper never mentioned race. Nicholas Wade, who never met a gene he didn't like, in the New York Times says, on the other hand, this study identifying main populations nudges scientists back onto the terrain of race. I'm going to skip this because I want to get to a major point. Nicholas Wade is not my point, this is. This was a computer program called Structure. And you put into a database thousands of people, in this case 1,075 people, put their DNA in, and then you tell the program to find patterns which are different for different groups. And with 3 billion base pairs and 3 million points of difference for any two people, a supercomputer will find patterns if you tell it to. And after considerable manipulation, the structure program found these patterns. They said that microsatellite markers are different for this group of, quote, people from Africa. This is the European SNP pattern, West Asia, East Asia. Now you see it's not definitive, but you can see there are patterns. In reporting out these data, they said, therefore, you can use Ancestry. But suppose you told the program to get 2 kinds of patterns. The structure program would do the following. Same data set. It would say that there are 2 kinds of people, the purple people and the beige people. That's it. Tell that program 4 kinds of people. It'll give you 4 kinds. And suppose you said you wanted to look at 14. Here'd be your pattern. And the work of Deborah Bolnick of the University of Texas, Austin, using the same structured program on the same data set, concludes that 14 is the best fit of all, given the criteria of the structured program on how to sort microsatellites. So now I ask you, why didn't they report that there are 14 different kinds of people? Why did they report there were 6? And the answer is not just rhetorical. It's that 6, or actually when Wade got a hold of it, 5 happened to coincide with pre-existing taxonomies of race in the culture. The genetic data said there are 14. The social data said 6. The media said 6. And ever since that was published, it's been called, by some people, the molecular reinscription of race. Back to my original slides. What are these ancestry informative markers? How did they come to represent Africa, Europe, Native America? What's going on here? And what is, after all, the claim? Point number 1, not at all technical. There's no such thing as a marker that's unique to a racialized population. All markers. Duffy's one exception in the literature, but even that is problematic. All markers appear in all racial populations. Therefore, what they're talking about is the frequency of the markers. That's important to remember. It's the frequency of the markers. There's no markers in any population that's defined racially. Furthermore, these are markers. They're not genetic functions. Just a quick side, this is all being taped, so I better be clear about what I mean here. 90% of the genome is in the non-coding region. 10% is coding, and those are called genes. These markers are almost universally in the non-coding region. They are not genes. Okay. They've been selected because their frequencies differ by at least 30% in these different population groups. So you tell the computer, I want to find a population group, 30% of this that's not over here or here. Okay? Now, I'll give you an example in a moment. But this is technical enough for you. I want you to make sure at least I get the basis covered before I end and give you an example of why this is smoke and mirrors. Admixture percentages were generated by formulas that compare frequency of aims between Europeans, Africans, Native Americans. Remember, it's frequency, not yes or no. These are now being used in research all over the country for what's called admixture mapping. All those in the defined group are tested for a panel of alleles and the frequency of which each marker appears. They then compare the frequencies between the 3 groups to come up with the values, and they're called admixed samples. Okay. Recommended reading, and then I'm finished with a rhetorical example. I'll leave this up on the board for those of you who want to pursue this conversation. But let me end with the following exercise. Let me take this group of people and call you Group A, and call all in the middle Group B, and all in the Group C. I'll then take your DNA and put it into a supercomputer, Blue Gene by IBM. Use a structure program, and I'll tell the computer, find differences in the SNP patterns of Group A at 30% frequency that are at that level of difference between groups B and C, that are not in these group populations at the same degree. The computer will find those differences, and you'll be Group A. Okay? That's your self-description. Group B, I'll tell the computer the same thing. Find differences, allelic frequency patterns, in Group B at 30% that are not the same in Groups A and C. And the same for Group C. I'll tell you the same thing. And you call yourselves Group C now. Now, you could reasonably conclude that that kind of research was nonsensical. Why do that? Unless Group A was all white, Group B all Asian, and Group C was all black. Then you'd say the computer had found something. The computer had found racial differences. You had inscribed in the computer structure program capacity for the reification of race. Now let's go back to that early slide I gave you. Send in your DNA to a company called DNA Print Genomics and get an ethnic estimation of your background. What are they doing? They start with the phenotype, all white people, all black people, all Asian, all, quote, Native American. They tell the computer, find frequencies, markers, distinctive at 30% that are different in those groups, and then this becomes the all-white prototype. Now, statistics, you don't need to be an expert to know that if I say you're 85% of something, I must know what 100% is. Yeah? 85% white? What's 100% white? How'd they get there? They used the phenotype, used that, and then said, find frequencies in the structure program, or whatever they're using, doesn't matter what the program is. That becomes the 100% white group. You send in your DNA to my company off the street, and I'll say, oh, you're 85% of that group 15%, and of group over here, you're a zip percent. How can you tell someone that they're not Zulu when you haven't sampled a Zulu population of the year 1500? You can't. In other words, the claims that are being made in sober circumstances on PBS about ancestry have no valid basis. And yet we nod assent. When someone says, you're not a single DNA percentage that's Native American, and my colleagues nod their head, I have to nod my head. Thank you.